Module 13, Oxidation and Antioxidant Needs. What is oxidation? Oxidation refers to the loss of an electron or gain of an oxygen molecule, while reduction refers to the gain of an electron. So this all is functioning at the atomic level as far as this topic is concerned. And generally speaking, we refer to all of the processes that we're about to talk about as oxidative reduction reactions and how antioxidants can combat some of the issues associated with the shuffling around of electrons. What is a free radical? Free radicals are basically the bad guys. This is when you have a molecule that's either gained or lost an electron in the outermost shell, and it basically causes an uneven pairing of electrons. So what ends up happening is that this atom now becomes very, very unstable, and it wants to become stable, so it basically reacts with substances around it, trying to steal electrons away from other structures, and so that causes damage. So we want to make sure that we mitigate this problem of these free radicals and try and stabilize them as best we can so we can get rid of them. So as an example here, I've got the nucleus of an atom. I've got the first orbital shell with a couple of electrons, and then there are 8 electrons here. This is supposed to be oxygen. It's supposed to have 6 electrons, so pardon the additional electrons there. But the point here is that if we have a atom that has an even pairing of electrons in the outer orbital shell, then it is stable. So it's not going to react adversely with structures around it. If we suddenly now add an electron to this molecule, or excuse me, atom, then it's going to be very, very unstable and turn into a real bad guy and start to negatively affect structures around it that it comes into contact, because it's trying to stabilize itself by gaining some electrons and stealing those electrons from other structures around it. So it's very highly reactive and is now considered a free radical. So what types of free radicals are there? Now, for all intents and purposes of this course, I don't want to get too in-depth into all of the specifics for each of the free radicals, but just understand that when you're reviewing the scientific literature, you may see variants of these free radicals and may be wondering, why do we have so many? And that's because there are different classifications for these free radicals. So the first of which are reactive oxidative species, which can be abbreviated with ROS as the acronym. There's superoxide anion radical, which is basically an oxygen molecule that has gained an electron. And most of this is coming from the electron transport chain, simply because oxygen reduction, if you recall, in the electron transport chain, you need oxygen to accept that final electron. So that reduction isn't always complete, which then ends up producing from free radicals. So this is a normal part of respiration and producing ATP via oxidative metabolic pathways. It's not very reactive, so it's not going to cause too much damage. Then we have the hydroxyl radical, which again is just a free radical that has this extra electron that's interacting with hydrogen peroxide here, and then you get the hydroxyl radical formed from it. It's usually going to form because there's some sort of free metal ion available, like iron or copper or zinc. If these are available, then you may get the formation of these hydroxyl radicals. It's very reactive, so it can cause a lot of damage, but the good news is it doesn't move around much, so it stays pretty localized and we can contain it a little bit easier because of that. Then there's the hydrogen peroxide radical, which is very mobile and can move about very freely. It's very reactive, so we need to do something about this free radical or it's going to cause quite a bit of damage. So it is produced by superoxide dismutase, which is an enzyme that is released in response to the immune system. So if we have any immune responses, superoxide dismutase is going to be released, and then it's going to be produced, or it will produce hydrogen peroxide radicals. Then we have the singlet oxygen, which again is just a free radical that can essentially borrow electrons from some sort of high energy release like radiation or oxidative stress and move the electrons to a new outer orbital shell, a much higher orbital shell with lots of energy in it. So it can cause some significant damage as well. Then there's the hydroperoxyl radical, or the .ROOH here, which is formed via chain reaction. So there's a series of reactions that occur in order to produce the final hydroperoxyl radical. Where do we get these free radicals from? Where are they coming from? Well, there are several sources, one of which are exogenous sources. So polluted air is a classic example. Cigarette smoke, being in that kind of environment. If you're in a city where there's a lot of smog, this is an exogenous source of free radical. The UV light of sunlight is a type of free radical energy. I mentioned the smog, the ozone layer. Lipid peroxides are all sources of free radicals. So in other words, cholesterol that has been oxidized before we ingest it can represent an exogenous source of free radical. We also have endogenous sources of free radicals. These are byproducts of normal cellular metabolism. So just by the very nature of being alive and running chemical reactions in the human body, we're going to produce free radicals. There's no way around it. The electron transport chain leakage, that's a source, as we mentioned earlier, of free radical production. Xanthine oxidase, which is an enzyme system, is going to produce hydrogen peroxide radicals. We have the cytochrome P450 system, which produces the superoxide anion. This is a system that is a part of the way that we detoxify things in the human body. There's the immune system that actually takes advantage of free radicals to combat some of the antigens, bacteria. And so as a consequence of that, the immune system is going to produce things like superoxide anion. There's superoxide dismutase, another enzyme that is going to mitigate some of these free radicals and cause some free radicals to be generated. The availability of iron and copper and other transitional elements, these are highly susceptible to hydroxyl radicals via the transformation of superoxide anion in what are called the Haber-Weiss reactions, where basically these electrons are being shuffled around and the elements actually can facilitate the production of free radicals. What is lipid peroxidation and its significance? Well, if you think about the phospholipid bilayer has a lot of unsaturated fatty acids within that phospholipid bilayer. And basically, they are very susceptible to free radical generation. And so we can take, if you take a look at the picture here on the right-hand side of the screen, we can take a nice unsaturated fatty acid and expose it to some free radical, and that will then cause damage to the PUFA. And because this PUFA is damaged now, it actually spontaneously rearranges and interacts with substances around it, and you end up producing some byproducts which aren't very nice. So aldehyde, malondialdehyde, and aldehyde in this form here can all generate problems as far as free radicals interacting with structures around it. So it's a damaged substance now that presents the possibility of further damage from having these lipid peroxides around. So just by having the phospholipid bilayer around and having those unsaturated fatty acids, those polyunsaturated fatty acids available, they are susceptible to free radical production and damage, and thus will further the increase in free radicals available. So how do we protect the phospholipid bilayer? Well, thankfully, we have vitamin E that does come in and actually donates its electrons to stabilize the damaged unsaturated fatty acids. So by donating the electrons, vitamin E now stabilizes that highly reactive oxidative or that highly reactive free radical and stabilizes it so it doesn't cause further damage. The problem here now is that vitamin E now is damaged. So vitamin E turns into a free radical, but the point here is that it is much less reactive as a free radical than the original free radical it attempted to attenuate. So then vitamin E is going to be fixed essentially by vitamin C. So vitamin C now is going to donate its electron and stabilize vitamin E and fix vitamin E. And so in that process now we've made vitamin C a little unstable and it's a free radical, but it's much easier to contend with this damaged vitamin C than vitamin E or the original polyunsaturated fatty acid that was damaged. And then finally, glutathione, which is a protein, is going to stabilize vitamin C and donate an electron to the cause and make that vitamin C whole again, and it is no longer a free radical. So glutathione is very, very easy to fix, and eventually it will get fixed, as we'll see in a few slides here. So why should I be concerned about free radical production? If this is a normal part of everyday chemical processing, why should I care about this, especially if I'm an athlete? Well, the problem here is that a free radical is potentially damaging to molecules around it. And so things like DNA, when you start making manipulations of DNA, that can cause errors in replication of cells as we grow older. So that can cause problems down the road. It's actually one of the theories of why we age. And that we introduce these small errors that eventually over time add up to bigger problems as we're older. Lipids are damaged, proteins are damaged, carbohydrates can get damaged. All of these spell damaged structures that no longer retain their original function and aren't functioning optimally. The other concern here is that this can lead to cancer and cardiovascular disease and even some nerve degenerative disease. So we want to be concerned about free radical production. We try to mitigate free radical generation and ameliorate the effects of these free radicals. So does exercise increase free radical production? The short answer is yes, absolutely, particularly if we're engaging in very high intensity, sustained aerobic exercise. And it's believed to be from the increase in oxygen presence than utilization within the mitochondria. So it's all about the mitochondria and what's going on there with that oxygen being the final electron acceptor and the production of free radicals. So this may actually cause an imbalance between the amount of free radicals available and the available antioxidant system defenses. So the good news is that as an athlete trains, as we train, we'll naturally increase our antioxidant defenses to combat the generation of free radicals. So that is the good news, is that there is an adaptation that can occur to mitigate these increases in free radical particles that are being produced from engaging in exercise. This brings us to our antioxidant defenses. So what are antioxidants? Well, as you may have already figured this out from talking about vitamin C and vitamin E and glutathione, these are compounds that essentially protect us from free radicals by neutralizing the free radicals, by stabilizing those free radicals from doing further damage. And so they act as scavengers in some cases for free radicals. They also reduce the catalyst for oxidation, so the rate at which we produce free radicals can be slowed down by having more antioxidants around. We can also repair the damage from oxidation. And we can bind up free metal ions so that those free metal ions aren't interacting with free radicals and then causing further damage. So it's all about balance of free radicals and antioxidants. And as we exercise and begin an exercise training program, we do offset this balance, but we can certainly bring that back into balance with antioxidants. So what are the different types of antioxidants? Well, we have proteins that can act as antioxidants. We briefly discussed one already, so glutathione, also known as GSH for short. And these are essentially amino acids that have come together to form this protein. We have glycine, cysteine, and glutamate are the amino acids that form glutathione. And they help essentially to regenerate vitamin E and vitamin C to make them whole again so that those damaged nutrients, those damaged vitamins are no longer posing threats and can further act as antioxidants. So you're removing the H2O2 by transforming glutathione into glutathione disulfide, or GSSG. And this is actually a reaction, if you look at this little picture here on the right-hand side of your screen, that requires the mineral selenium. And there's also an enzyme here, glutathione peroxidase, that facilitates this reaction. So here we have glutathione at the bottom here with some hydrogen peroxide, and via glutathione peroxidase and selenium, we can convert that into glutathione disulfide and release a couple of water molecules to get rid of the free radical here, and the free radical being the hydrogen peroxide. So we also have quinones, which the most common one that you may have heard of is coenzyme Q10, and this is involved in vitamin E generation. It's also protecting the phospholipid bilayer, and it scavenges for the peroxyl radicals. Now, in the literature, you'll notice that there are some mixed results as far as coenzyme Q10. There are many studies that show that there's tremendous benefit from having coenzyme Q ingestion. Then there are other studies that basically show that there's no effect, which then brings me to the statement down here. So its role as an antioxidant in healthy status, healthy adults, has not been substantiated. So you'll find the literature to be a little controversial with respect to the coenzyme Q10 and the quinones. There are enzyme systems. I mentioned glutathione peroxidase in the previous slide. So again, this is part of that conversion of glutathione to glutathione disulfide, and again protecting us against free radicals and mitigating that problem of having these things hanging around. There's glutathione reductase as well, so that we can convert glutathione disulfide back into glutathione. So we want to be able to regenerate glutathione, and this is the enzyme that is going to be responsible for doing that. There's also superoxide dismutase, or SOD. This is also very important in this whole system. It's going to accelerate the conversion of superoxide so that we can, again, get rid of these free radicals. We do need some minerals for this particular enzyme to work properly. Manganese is one of those minerals, as well as some copper and zinc in order for it to work properly. Then there's catalase, which is located in peroxisomes and perhaps even myocardial mitochondria. And again, we need this enzyme so that we can facilitate the proper functioning of these reactions. So iron is a good example of this, so that we can properly utilize iron and not allow it to interact with free radicals and cause further damage. Then there's peroxidase, which is found on HDL particles. So its function is to protect HDL against oxidative damage and free radical generation. We also have vitamins, as we saw earlier. Vitamin E donates its electrons and protects the phospholipid bilayer, but it's also found in the mitochondria and adipose tissue, liver, and muscle tissue. But its main job really is to protect those PUFAs, those polyunsaturated fatty acids. Now, one interesting note is that vitamin E comes in a lot of different forms. There's tocopherols and tocotrienols, and it seems that the alpha tocopherol is the most potent out of all of the vitamin E forms. Vitamin C, of course, as we saw, regenerates, fixes vitamin E by donating an electron. Again, this is all in an effort to reduce the damage from free radicals, and vitamin C now becomes a less damaging type of free radical. And it's interesting that a lot of the literature you'll find also revolves around cigarette smoking and protecting lungs from free radicals. So there's quite a bit of work with vitamin C as far as an antioxidant in protecting the lungs. If you consume high levels of vitamin C, it actually can turn into a pro-oxidant with very high levels of iron. So we do have to be careful with that situation as well. Vitamin A has been studied for its antioxidant properties, but it seems that it's kind of a weak antioxidant. And if you overconsume vitamin A, especially in its active form, so retinol, retinoic acid, so that would be the top three forms of vitamin A, the active form that you see in this figure here, that can actually be toxic to us in high doses, so we do need to be careful of that. Beta carotene, as you recall, is a pre-vitamin, so it's a precursor to the active form of vitamin A. Minerals, we saw selenium that was needed for glutathione peroxidase to function properly. This slide is a nice slide that kind of pulls all this material together. So I'm going to walk you through this slide, and let's start down here. Well, first let's orient ourselves to what we're looking at here. So this area here is basically the phospholipid membrane. So any plasma membrane is this area. And then, of course, this space below, this is the intracellular space, so this is inside the cell. This is all happening inside the cell. So if we start down here with substrates, so substrates are any substances that basically free radicals are going to attack. So it could be DNA, it could be protein, could be a carbohydrate, etc. So substrates interact. They may interact with the cytochrome P450 system, and they may produce a free radical. And then minerals that are available here to help superoxide dismutase then can convert that free radical into hydrogen peroxide. That hydrogen peroxide then can, via catalase and a little bit of iron here, be converted into water and release oxygen to relieve the body of some hydrogen peroxide. Now we can also take that hydrogen peroxide and convert it into water via the glutathione peroxidase enzyme and selenium that we talked about earlier, and taking glutathione and converting it to glutathione disulfide. And then to make glutathione disulfide whole again, we take NADPH and take off an electron via glutathione reductase, release NADP, and then we have glutathione once again ready for action. We can also take the hydrogen peroxide and via peroxidase and some minerals here, release some water and produce some free radicals that are less damaging, which then can, via vitamin E, can actually be converted into water. Again, so we're trying to take these free radicals and simply convert it back into water, something that we can actually use. So all three of these free radicals here can interact with vitamin E and be saved basically from damaging us and converted into water. Now, some of these free radicals can come back and interact with PUFAs, unfortunately, and then, of course, cause those lipid peroxide radicals, which cause further damage. So it's essential to have vitamin E around in order to mitigate these free radicals. Now, vitamin E, of course, in this whole process can become damaged. So then vitamin C comes in, and vitamin C is now damaged, but it's a less damaging type of free radical. So the end result then is going to be taking glutathione and saving vitamin C from free radical damage. Now the top of the screen here, of course, when we ingest dietary PUFAs, those are being incorporated into the phospholipid bilayer, and so vitamin E plays a very important role in protecting that PUFA from free radical generation. So as far as other antioxidants, we have phytochemicals. Now, phytochemicals are simply plant chemicals. It could be the pigment in a plant, could be some structures in the plant. But the important point to remember here is that these are not essential nutrients. So we don't have to eat them in order to survive, but they do provide us with salubrious effects. That is, they provide us with health beneficial effects. So that's an important distinction to remember. The phytochemicals are not essential to staying alive, but they do provide us with some health benefits. So carotenoids are a type of phytochemical that are found in things like tomato products. So lycopene is found in tomato products. There's beta-carotene, alpha-carotene, lutein. These are all carotenoids. So they're pigments that give color to the particular food item that you're talking about, the particular plant item. And they're beneficial to us because they can act as antioxidants. We also have glucosinolates, which again are functioning as antioxidants. They're found in cruciferous vegetables, particularly the brassica, like broccoli and cabbage and cauliflower. We have isoflavones, which are found in soy foods, which again are acting as antioxidants. We have the isothiocyanates, which are found in cruciferous vegetables, the brassica group in particular. Again, pigments that can be used as antioxidants. Lignans are also an antioxidant. These are found in things like flaxseed oils and soybeans and nuts and berries and rye and bran. Again, they're functioning as antioxidants. They're not essential, but they're functioning as antioxidants for us. We have organosulfide compounds, the most heavily studied one of which is garlic. Garlic is very rich in these organosulfide compounds, and there seems to be a lot of work as far as trying to reduce the risk for cancer and that cancer development coming from the free radical generation. So it's functioning as an antioxidant. There are phenolic compounds, which happens to be the largest class of phytochemicals here, but we've got phenolic acids, we have flavonoids, we have catechins from things like green tea and anthocyanins. These are all functioning as antioxidants. There are phytosterols, which are the plant form of sterols, and so these are also functioning as antioxidants. Things like vegetable oils have lots of phytosterols in them. Then there's resveratrol. If you like red wine, you like grape juice, you're going to get some resveratrol. Even peanuts have it. Again, functioning as an antioxidant. There are saponins, which are found in things like alfalfa sprouts and potatoes and tomatoes and ginseng. Again, an antioxidant here. Then there are tannins, which are found in things like black-eyed peas and grapes and lentils and red and white wine and tea. Again, antioxidants. Then there are the terpenes, like limonene and carvonine. These are coming from citrus fruits and peels and cherries and ginkgo biloba. So the theme here is that we have these phytochemicals. They're coming from plants. So there are all these chemicals in plants that if we ingest the plants and the products that they're producing, then we're going to have higher antioxidant defenses because of all these phytochemicals that were ingested. So we need to eat our fruits and vegetables, essentially. One of the special notes here that I think is really important going along with what I just finished saying is that, when we ingest fruits and vegetables and things like garlic, etc., these substances, these plants have thousands of phytochemicals, and there are lots of combinations of these compounds. And one of the things that I'd like to impress upon you is that when you're evaluating the scientific literature, it is very difficult to isolate these compounds. So if you're saying that, the increasing the consumption of fruits and vegetables provides antioxidant defenses, and then suddenly, you're saying it's because of this one phytochemical, that becomes a bit of a stretch because you really have to study just that one phytochemical to determine what the benefit of that phytochemical is going to be. And even then, is that realistic to be consuming that particular phytochemical by itself? Does it make more sense to have the phytochemicals ingested in food as a whole? So that's something that the scientific literature does struggle with quite a bit, and you'll see that, and that's why there are a lot of controversial studies in this area of study. As far as athletes, do athletes need antioxidant supplements? Well, the current recommendation is that we really don't need to be supplementing the athlete with antioxidants, so individual supplements of antioxidants. We'd prefer to encourage the athlete to basically consume food sources of antioxidants. So making sure that the athlete is consuming those fruits, those vegetables, etc., and getting the antioxidants that way. If there is, for whatever reason, let's say you have an athlete that's susceptible to URTI, so upper respiratory tract infections, then you may want to increase vitamin C, for example, in the diet. But again, that's on a case-by-case basis. But as a general recommendation for all athletes, that simply isn't there at the moment. So are there any risks to supplementation with antioxidants? Well, we know that vitamin D can actually exacerbate autoimmune and immune system diseases like asthma and allergies and diabetes and rheumatoid arthritis. So we do want to be careful there, and again, case-by-case basis. Vitamin C can actually promote kidney stones at high levels. It can increase the risk of iron-induced cardiac failure. And if you have a person that has hereditary hemochromatosis, where these folks actually absorb too much iron, vitamin C actually enhances the absorption of iron from the gastrointestinal tract, and so that could actually cause liver failure and liver damage. So we want to be careful in those individuals. Beta-carotene, actually in some studies, actually found to increase the risk of lung cancer and even cardiovascular disease.